Hello, my name is Alexei Lopez Herrera, and today I'm going to talk about the co-immunoprecipitation and the immunobloating. This is part of the Group 2 assignment for the presentation. Okay, let's get started. Let's get started first with the introduction and the principle of techniques for both the co-immunoprecipitation and the immunobloating. The Western blotting or the immunoblotting, which is the same thing, is a common technique to detect near levels of protein expression in the cell or tissue extract. This actual technique measures the protein levels in a specific given biological example through antibody binding to a specific protein interest. And I put immunoprecipitation here because it's so similar to co-immunoprecipitation when it comes to the end result. So the immunoprecipitation is an existing common technique used to enrich a near-specific protein from a heterogeneous cell or tissue extract utilizing a certain target specific to the antibody itself. And now when it comes to the co-immunoprecipitation or the co-IP, is the pull down of the intact protein complexes, which the IP, which stands for the immunoprecipitation, and the co-IP, which stands for the co-immunoprecipitation, are valuable and widely used techniques to identify protein-protein interactions and novel members of protein complexes. Now let's talk about the application of techniques. And when it comes to the application of techniques for the immunobloating technique is that I divided it in three general parts, which is the validation, the controls, and the protocols. Let's get started with the validation first. When it comes to the validation of the application of techniques for the immunobloating technique is that antibodies are recommended at optimal dilutions using specific buffers and protocols. It's pretty important to do such steps. The second part is the controls, which for the immunobloating, which is it is that is recommended to use positive and negative controls that cells extract so that the antibody performance can be best tested. It's really important to keep that in mind. And finally, let's talk about the protocols when it comes to the immunobloating for the application of techniques, which is it's really crucial and important to read the protocols with such full detail in order to start the experiment quickly and to minimize time and the reagent waste overall. So it's really important to actually waste no time at all in doing this kind of experiment and actually minimize the time and actually to get the best results as possible. Now let's talk about with the application of techniques when it comes to the co-immunoprecipitation, which is the high degree of the antibody specificity coupled with the availability of thoroughly validated IP beats and buffers to ensure reliable IP and co-IP, which both stand for the immunoprecipitation and co-immunoprecipitation data to elucidate protein-protein interactions and protein complex compositions as well. Okay, and now let's talk about the two pictures that we're seeing here right now. And the picture on the left hand side, it's about the co-immunoprecipitation, and the picture on the right hand side is about the immunobloating technique or the immunobloating study right here. The left hand picture is the co-immunoprecipitation, right hand picture the immunobloating. Okay, first let's talk about the picture on the left when which I said earlier is about the co-immunoprecipitation data. And this and the co-immunoprecipitation data that is actually showing the pull down of the mTORC1 gene complex using antibodies targeting various complex components, including the G beta L and the mTORL, OR, and the raptor. The data was generated for something called the HeLa cell extracts and the certain antibodies, which it's right here. Now the right-hand side picture, which is about the immunobloating studies. It's about or it's also called the Western blot, which extracts from various cell lines, untreated and treated as indicated with the LY294002 gene, which was used with a kinase inhibitor, which is a human insulin-like growth factor, which activates the AKT via the IGFI1 binding, or the human platelet-derived growth factor AA, which is this picture right here of the immunoblotting. Okay, finally, let's talk about questions that I had. Three questions that I came out with is that if can someone use the exact same antibody for the co-IP or the co-immunoprecipitation and the IB, which is the immunobloating? The second question that I came up with as I was reading between the co-immunoprecipitation and the immunoprecipitation is, can I use more than one control medium in both the co-immunoprecipitation and the immunobloating in the same time? And finally, I came to the final conclusive question that I had is, can I use a control medium without any of the antibodies? So these are the three questions that I came up with as I was reading between the co-immunoprecipitation and the immunobloating cell division techniques. Okay, and this is the end of my presentation. Thank you for listening.